Our first speaker is Dr. Paul Vincelli, and he is going to talk about advanced concepts in fungicides. So with that, let me hand this over to Paul.
Thank you, Rick. And folks, thanks for attending our Extension IBM update. And I'll do my best to give you some information on the basis of the title. But I noticed that I put part one in there because there's really about 15 or 20 different topics easily that could be discussed at future dates if you're interested in some of these advanced aspects that hopefully will help you understand how to use fungicides and other pesticides better. So maybe it's important to start, depending on the audience, with this slide, and this really illustrates why people use fungicides. I think many of you know why people use fungicides, but this was the damage from an infectious disease of perennial ryegrass throughout much of the eastern U.S. This was natural inoculum, and this is what it did to these golf courses and other landscapes. Just clobbered it. And I don't have a picture because we didn't really understand this disease in 1995. I don't have a picture of what fungicides would be capable of doing in this collection, but if you could, well, I'll show you some data. Maybe that's the best thing. All right, so this was in 1998, 3 years after that ferocious epidemic, and these were the levels of disease through the summer, and you notice that the blue line goes up to nearly 100%. That represents pretty much what you see here, and so the water control was the blue line was representing that disease level going, skyrocketing to almost complete death. And those other two treatments were treatments with two or one applications of a common fungicide. And they did a beautiful job of controlling the disease for most of the summer. And so this is why. I mean, people were losing jobs over this disease. And so if they could save their golf course and save their job, of course, with a legal product and a safe product, and many people opted to do so. Now, I think one really important topic to discuss, and the focus of this talk, is biological fungicides for disease control. This has really become a thing. Basically, people are very aware of the benefits and risks of synthetic chemicals used for disease control, but at the same time, there's a desire even among people not in agriculture to see more biological products being used for disease control, and I'll talk about some of the advantages and disadvantages in a moment. But it's probably good to understand at least the basic concept of what's going on when we either create or use a microbial or microbiological product. And so you can see that there are these strands of fungal growth at this high magnification, and some are labeled R and some are labeled T, and then the R is the pathogen in this disease, and the T is the fungus that attacks the R disease, the R fungus, the Rhizoctonia fungus. So the T, right, that strand in each slide or each image is coiled around one of the strands of the pathogen. And so that coiled fungus is actually attacking the pathogen, yay, and it's going to suck it of its nutrients and kill it. And that's kind of a nice illustration of what happens with microbiological types of products. This is something akin to what you'd see if you looked in the soil or on the leaves. Okay, and this is just another example of real infections by biological products. So maybe we'll start with the one on the upper right, and you can see that there's this fungal filament, this growth, but there's also these little beads. They're oblong kind of beads. Those are the bacteria that is the product that kills the fungus. Okay, so you get these really interesting interactions that are quite beautiful to our eyes at these microscopic levels. But the important thing is that the biological agent is interacting with the fungus that causes you disease problems. And through that interaction, the biological agent is able to kill or suppress, at least, but usually kill on some level, the fungus that's causing disease in your crop. All right, so this is, again, to give you a sense of what's going on with these materials. This is actually a close-up of a leaf surface, and you can see there's these kind of small valleys on the leaf surface. There's a lot of those, but then there's these very, very tiny beads, that are kind of circular in structure. That is the biological agent in this case. And so you can see, or no, sorry, no, not this one. The beads are on the fungicide that would be used. This is the chemical fungicide. And so you spray it and hopefully get this very uniform coverage like you see here, so that these little tiny beads of the fungicide are there to do their thing when a fungal spore lands on this leaf and gets the moisture that it needs to grow. Okay, and now this is the biological agent that might be used. And so, first of all, you're looking at a stoma, which is the opening to the inner parts of the plant and allows gas exchange. And then within that stoma, you can see on the edge, they almost look like the lips of the stoma. And inside the stoma, you can see these bead-like structures, oblong bead-like structures. And that is the biological agent. And then this slide, it's sitting there waiting to do its thing. So when a fungal spore that attacks the plant comes along and starts to grow near this opening or even into it, then that's when the biological agent is going to continue to multiply and attack the fungal strand and hopefully kill it. So it is the same concept. This is a chemical fungicide. This is a biological fungicide. They're both fungicides. They both kill the target. And so in some senses, there really is no difference between these two. And in some cases there are. Also, as we look on this, one of the things you can see is this concept of number one on the right. Prior colonization may be beneficial. So if you're using a biological agent, you want that thing there in advance of the infection, okay? And sometimes that means getting out there and spraying even before the infection has started. But that's what you often have to do with these biological agents is get a particular timing that makes sure that they're there when they're needed. This is usually as a preventative in advance of infection kind of thing. So and then the other thing that this illustrates is that the systems that the biological agents and their formulated products that are used for disease control, they're really simpler in concept and in function than chemical pesticides. A chemical pesticide, you spray it, it goes into solution in the dew, it poisons the cell when the spore starts to grow, and that's it. But if you are using a biological agent, now just look at this picture for a moment and realize that this bacterium needs to be growing and actively so. It needs to be growing by the time the spore gets there. And you may or may not be able to achieve that. Maybe it's the weather is not conducive to the biological agent. So there's going to be some complications in using these biological fungicides. All right, and to remind you of infection, and this is what you're trying to prevent with these biological fungicides, the pear-shaped structure, kind of in the middle there on the left-hand side, that is the spore. Okay, and then at the tip of that, you see that there has emerged a structure coming out of the spore that's not coming out of the plant, it's coming out of the fungal spore. And then it produces this pressing structure onto the leaf surface, and that's how it penetrates and infects, okay? So that's the target. Imagine that's where you need your biological agent, is where infection is happening. So, you might think it's important to treat preventatively, and you'd probably be right. Okay. All right. These are typical degradation curves for pesticides in soil, on leaf surfaces, and so on. So there tends to be this rapid degradation initially, and then it tapers off and, eventually slows down to the point where there's not much happening on the leaf surface. That's typical for chemical pesticides. With biological pesticides, this is a more typical example of what you'll see. And the main thing to notice is you get ups and downs and ups and downs. And because it's growing at some times and under some conditions. And then the biological agent may not be growing at all under other conditions. So you're going to get ups and downs from the biological pesticides. And that's something that affects management too, because if they're going up and down, they may not always be in a state where they're able to give you the disease control you want. Okay, this is one of many, many review papers in the scientific world on biopesticides, biological pesticides, and so on. And there is a lot going on. It's really become a trend, and that's a good thing. But you notice the authorship of this particular review. I think they're all from India, and I have reviews from other parts of the world as well. And what this illustrates is that a lot of this work and a lot of the vision for this work really has come from other countries and many of the developing nations, probably for the reasons of not wanting or having the funds to spend on the synthetic chemicals quite so much. But the developing nations have been in the lead on this in a big way, and so we're all in this together and learning from each other. And I'm glad for the work they've done, and we're working on it too. All right, I've got several of these tables that define some of the benefits and risks and the differences between chemical control, as you see in the middle column, and the right column, and for biological control. And maybe the most you can read these yourselves, but I think what is most important is towards the bottom, where it says risk of resistance, large risk, and very small with the biologicals. There's a lot on this table I agree with, but I'm not so sure that that's going to hold up. Maybe it will. Look at the one below that, the specificity. Okay, so what that means is how specific is this biological agent that controls disease? Is it going to be good against one disease, or against 20, or against 100? And if it's more specific, that targets fewer fungi, then usually that means it's going to be it's going to be subject to resistance buildup. So I think that I do agree with most of this table, but I do think that if there is if the specificity of the product is very limited, so it maybe only targets 1 or 2 or 3 or 5 fungi, then we may see resistance develop in that situation. It's a prediction. It could be wrong. We'll see as we go through time. All right, some more comparisons between conventional and biopesticides. Let's see. The good ones to talk about here. I wish I was there. I'd ask you which ones you wanted to hear about, but I think that here, let me give you an example of one I do agree with, so you don't think I'm just trashing these nice papers. Here towards the bottom, it says conventional chemical pesticides are known to be contaminants of water and soil, and the thinking is that the biopesticides do not pose a risk like that in terms of contaminating water sources. And I think that's probably true. We're going to be surprised to find contamination of water sources from these biopesticides. But again, we'll keep our eyes open to that possibility. Let me look one more time back here, developmental time. Okay, that's good. All right, so, and we can come back to that if we have time. So here is an experiment we actually did at the University of Kentucky, where we created a compost tea, which is basically it's not really a tea from heated water, but from room temperature water, And you just take compost and you mix it with or without aeration, with or without extra nutrients, with water, and you take that extract and you spray it on plants, and some people report disease control, at least on some level. We tried to do that kind of work, and I think we did some pretty nice experiments. And on the right is the percentage of rhizotonia damage on our experimental system, our plant system. And we used 4 different compost teas, and they were all equally ineffective. We used 3, I'm sorry, and they were all equally ineffective. And I think really maybe I'll just jump to the next one. There's more data showing basically the same thing, that the compost teas did not provide any disease control, but or very, very modest disease control, versus the chemical fungicide, which provided excellent disease control. And again, I'm not really blowing the horn for chemical manufacturers, but that kind of thing convinces. They don't need to see advertisements. The growers will understand the meaning of this pretty easily, right? So why did we get results like that? And we can get variable results with biological materials. That's just the way it is. And so why is that? Well, there are 2 reasons I think that are worth highlighting. One is, so the leaf surface. Think about that. Lay on a hot putting green in the middle of summer during the middle of the day, and that the leaf surface is physically inhospitable for microorganisms. It's just unpleasant to be out there in the rays of the sun in those conditions. So microbes don't have they don't always have the best opportunity to colonize the leaves and protect the leaves. But what about the soil? Now, the soil, of course, is very friendly to microorganisms because it's usually moist. Temperature is much more manageable. But the problem is that microbes like that. And so not only your microbe that you've sprayed likes that, but so do millions of other microorganisms. So there's a really intense competition among soil microorganisms to be able to function in these systems. It's not impossible. There are some very interesting products that are coming out and have been coming out from the world of biological control. And actually, some of the most promising ones, not all, but some are coming from the major. It took the major chemical manufacturers a little more time to come around to starting up programs on these kinds of things, but they did. They're working on it, and there's some interesting results that are coming out. So we'll continue to watch this. All right, I have just a couple more minutes here, so I'm going to look through. All right, this is a great slide in terms of learning potential. So let's go with it. This is dollar spot damage 8 weeks after the spray of a fungicide. And the left was sprayed with fungicide, the right was sprayed with water. And now, at the time, so I put up the question, which plot was treated? And you might think it's a trick question, and it kind of is. If you came back to this trial 3 days after treatment with a fungicide to kill dollar spot, the one on the left looked great, where you controlled dollar spot, and the one on the right was clobbered. But back 8 weeks later, and the one on the left is slammed by dollar spot, and the one on the right looks good. And we think, just to wrap it up, we think what's going on here is when we sprayed superconazole, we disrupted the natural biological control that is present in this soil, and made it come back worse over time. Okay, so I could say more about that in the Q&A, but I do want to stop there and take the questions that you all may have.
So do we have some questions for Dr. Vincelli about fungicides and biofungicides? This isn't a question, but I have to say bioinsecticides have taken a pretty good share of the market. So bioinsecticides have been a proven alternative.
Wow, interesting. I hadn't followed that situation. Yeah, that's good. I wonder if any questions on that topic. Rick, or you've got more experience, and interrupt me if somebody has a question. But while we're waiting, is the are the bioinsecticides, are they? Do you think that they're less safe, or sorry, more safer than the chemical pesticides?
I hesitate to use the label safe versus not safe, because all that depends upon the state of the science at the time. But I would clearly say that the bioinsecticides tend to be reduced risk materials compared to many of the synthetics. But again, I'm careful. I don't want to generalize and say, you know, all the synthetics are the same. They're not. There's big differences between products, and even some of the synthetic products are considered reduced risk materials by the EPA. So I don't want to generalize.
Okay, very good. Thanks.